one of the major role of Murburn concept to play a role is this drug metabolism and drug dose, etc. I think all of you might have convinced on that aspect. So to chair this, I can say the practical oriented session is Professor Sabita M. She is principal of the Amrita College of Pharmacy here. So it is my pleasure, and on behalf of the Swadeshi Science Movement and other organizations involved in this seminar, I welcome Professor Sabita and request her to occupy the chair. And Dr. G.R. Sridhar from Vizag, he was supposed to join us, but he will be joining only online. Yes, sir. Good morning and welcome. So, because of the, what you can say, weather problem, he decided not to come. So I welcome him, and we have discussed Dr. Suresh Nair, he will be joining shortly. He's on the way. Dr. Ashish Kandalwal, he's here already. I ask him, request him to occupy the seat. Then, yeah. And I also request Professor Venugopal of Calicut University to occupy the chair. Then we have speakers, Dr. Daniel Andrews from LDH cyclooxygenase. Cyclooxygenase. He's available. Please, you can be there. Okay. And Abhinav is, yes, Abhinav. So both of you, please. And then later on, it is up to you to decide where to sit. We'll initiate. And again, two more experts will be joining online. Professor Gabriele from Italy and Dr. Daniel from USA. Hopefully they will be joining. So with this, I open the floor to the chairman.
Dr. Murli.
Good morning, everyone. I thank the organizers for this opportunity to chair a wonderful session on drug metabolism, dose responses and medicine. The first session on the second day of this discussion forum. So I invite Dr. Abhinav Parashar to talk to us on CYPs and dose responses. Over to you, sir.
Sir, you're on. Sridhar, sir. Sridhar, Dr. Sridhar is a physician. He's an endocrinologist. He is the director of Diabetes Research Institute and also a fellow of National American Endocrinology Society and FRCS. So FRCP. And so he's a researcher and he wants to say a few words before the session starts. He's a co-chair with Professor Sabita. Thank you. Sir, you are muted. Sir, please switch on your mic.
Yeah.
Yes, please.
Good morning, Dr. Murli. The next best thing to being in person is being on Zoom. People like us, we are not as dry as Rajasthan, but still news of flash flooding and orange signals, they really put me off from being here. Thank you for arranging to have the Zoom meeting. When I hear of Murburn, I am reminded of the saying about Tolstoy's War and Peace. One man said, I really envy the person who has not yet read War and Peace, because I can't have the pleasure of reading War and Peace for the first time. So in the same way, the Murburn concept has had its firm foundations over the past 20 years, and now it has taken shape and it is being established as a very, I wouldn't say game-changing, it's going to change the way in which biochemistry is understood. And therefore, what I have seen yesterday and what I have realized today is there is a lot of work that can be done. The work that was established using the previous concepts and what you have initiated and what so many of the other experts are working is trying to apply the Murburn concept into a variety of biochemical fields. Had it been 20 years since, when it is already established, the researchers would have found it difficult to find an area in which to work. But here, this is a virgin field. I deal mostly with diabetes mellitus, and our association with reactive oxygen species is something very bad, quote unquote. That's because most of the complications are attributed to the adverse effects of reactive oxygen species. From the presentations, I have understood that ROS are not bad by themselves. Just as you can't say fire is bad because it will burn your hands. You can use it in such a way that it will explain a lot of things. So diabetes is one area where the complications, pathogenesis, all of them are related to insulin resistance. And the reactive oxygen species are one of the pathogenic factors. So for those of you who want to enter this area, it's a very important and inviting field. And second is we had worked on a protein called butyrylcholinesterase. Basically, it breaks down the drugs, toxins, as well as anesthetic agent when it is administered. We wanted to see whether it has any other roles. And over the past 30 years, we've done a number of studies in which the other roles of butyrylcholinesterase have been identified. I think of butyrylcholinesterase as a country cousin of the Murburn concept. And when I was going through the publications of Dr. Murli, I found that the Murburn concept can be applied to toxins. So there is a greater relationship between butyrylcholinesterase and the Murburn concept than merely the reactive oxygen species, insulin resistance, and diabetes mellitus. So for those of you who want to take a look into this, here is a molecule which can be explained or studied in terms of the Murburn concept. Having said this, I look with pleasure to the two scholarly presentations by the speakers. Thank you for the opportunity, Dr. Murli.
Throw the slides, please. Yeah. Very good morning to one and all. Okay. Sorry. Yeah. Very good morning to one and all. So today I'll be discussing a bit of the experimental evidences that actually led to the formation of what you're seeing today as Murburn concept. This is just some part of the experiment, and some other experiments will be the data from from other experiments will be shared with by my colleague, Dr. Dani, and further by Dr. Ellen. So I'm going to talk on the drug metabolism and dose response aspects, where the journey actually started. So, Sir Murli sir was talking yesterday about the MOX-4 system, that is the oxidative phosphorylation, which is obviously one of the most important things, respiration, that we have cracked down. And what you see over here on the left-hand side is the microsomal system. That is the microsomal xenobiotic metabolism system, metabolism on the left-hand side, which shares a lot of similarity with the oxidative phosphorylation. And this is what Sir had talked about yesterday. But then the journey actually started with the MXM system, the microsomal xenobiotic metabolism. So some of the key features that are shared commonly, you can see that the NADH requirement, and then the requirement of the substrate to give it a pull. I mean, the thermodynamic pull that is required based on our Murburn concept, the thermodynamic pull that is actually created because of the presence of the substrate is quite an important step. And then further the reaction happens over here. I mean, it's taken up. We'll talk about that in much detail in the coming slides. And a similar thing is observed in the oxidative phosphorylation. Now, yeah, so the thermodynamic pull that we were talking about, this is the experimental proof of the pull that that I have just spoken about. You can see these two different systems. These are peroxidation systems, which share a lot of similarity with the microsomal xenobiotic metabolism. You can see there is no reaction happening over here, and as soon as you get the substrate, which is ABTS in this particular case, and also here, you can see how quickly the reaction is driven. So this proves the role of thermodynamic pull in the presence of substrate. So as long as you don't have it, the DROS that we are talking about since yesterday is kind of moderated by the enzymes that we are dealing with. And once there is a substrate, the requirement of that DROS is very evidently seen here and is taken up. So yeah. Now let's get into the system a little bit, the microsomal xenobiotic metabolism system, the P450s, for that say, we are going to talk a lot more on P450s, and there will be some instances where we are going to see the peroxidase system. So here, what are the key players? You can see you need the electron donor. NADH or NADPH is one of the first thing that from where the reaction begins. I mean, these are some of the partners. So I'm also talking about the classical mechanism. Then we'll refute it. We'll show how that doesn't really work. And then then you need an electron transfer mechanism, which is done by the flavin containing enzyme, which is otherwise known as the CPR, which is a cytochrome P450 reductase. So this reduces the P450. This is the classical understanding. But basically, what we are trying to show here is that these are the components of the reactions. Okay. So, and of course, without the oxygen, nothing's going to happen, right? But then, what do we talk in Murburn? We say DROS drives the reaction, right? So when you're talking about DROS driving the reaction, and the reaction is happening at the SIP, that is for sure, right? So you don't, when you're talking about the DROS driving the reaction, the DROS can be generated here, or it can be generated here, and then can be transferred to the P450 system, right? You don't really need a relay system, a very deterministic relay system for that purpose. So, and yeah, of course, I just missed the cytochrome B5 is again a major modulator of the ROS. So when you look into the xenobiotic metabolism, you will see P450s are able to carry forward all of these kinds of reactions. Like you can talk about hydroxylations, you can talk about the nitrogen and sulfur oxidations and epoxidation, dealkylations, all these reactions and many more that it can carry forward. So the thing that I was talking about. Now we are just having a look at the classical understanding and the Murburn perspective. So when you talk about the classical system, the reaction begins with the binding of substrate. Now I'll go to the other slide. This is one way of presenting it. Okay, fine. So it actually happens when there is a substrate. See, RH is actually the substrate. So the low spin state of the enzyme requires the substrate to bind here, according to the classical mechanism. And then only after the substrate binds, it can, the whole electron flow can take place, and only then the reduction will happen, and only then there will be an activation of the oxygen. And eventually, all this time, you need the substrate to be bound here. And then the hydroxylation or the other reactions that we have seen might actually happen. Now, looking at all this, it really takes a lot of time, and it's not very fruitful way of explaining a very, this kind of a reaction. You get a very low turnover with this kind of an understanding, with this kind of a scheme. But then when you talk about Murburn perspective, Murburn concept doesn't talk about binding of the substrate here in the first. It talks about the formation of ROS and the modulation of ROS in the system here. This enzyme, that is P450, can generate ROS of its own. And of course, as earlier I have told you, it can also be generated by the flavin system, the CPR, right? Now, in further slides, we'll see the role of CPR as well. And then you have the modulators over here, like cytochrome B5. And now, as soon as the substrate comes in the vicinity and you observe this thermodynamic pull, there is the reaction. There's a quick release of this DROS, and it can just attack the substrate, give you the product. So this is quick. And the mild, unrestricted burning that keeps on happening. That's the main understanding of Murburn. The burning happens continuously, mildly. That's why Murburn, mild, unrestricted burning. It is continuously happening. And as soon as the substrate comes in, it is going to be released and the reaction happens. Now, it's the same thing presented in a little different way. And here, we will not call it the classical mechanism. We'll call it the erstwhile because we have very clearly refuted it. So we'll call it erstwhile hypothesis that you see in your textbooks probably. Now this is dependent on the substrate binding to cytochrome P450, which is not the case when you look at the Murburn concept. And then this mechanism requires the binding of not just the substrate to the P450, it also requires the binding of P450 itself to the cytochrome P450 reductase, that is CPR enzyme, like what we have seen here. See, this binding is required in the erstwhile hypothesis, where it's not really required in the Murburn concept. So, more facile, better, easier for the substrate hydroxylations or the reactions to actually proceed further. The product from DROS formation requires, okay, yeah. And what is the driving force here is the thermodynamic pull, not the binding of the substrate, right? So much more feasible. Thermodynamically, it is a much better explanation than the erstwhile hypothesis. Okay. Now, when you talk about the erstwhile hypothesis, it also mandates that everything happens in the active site, the concept of active site, the lock and key model, or maybe the induced pit model. Either of that talks about the binding of substrate in the active site. So unless this happens, and everything happens only here, right? So what do you expect when things are happening in the active site? You can get only one kind of a product, and you can only allow certain kinds of substrates. But when you look at the P450 system, you will see a lot of promiscuity. What is that? A lot of substrate, different kinds of substrates, right? Now, we'll come to that slide in a few seconds. When you look at the Murburn model gives you the freedom to understand the binding of substrate that can happen either here this is a substrate, xenobiotic is the substrate. We are talking about the xenobiotic metabolism. So it can bind in the active site, or it can bind somewhere near the channel, or maybe some other place from where it can allostatically modulate the enzymes reactions, right? So this gives you a lot of freedom to explain the kinds of observations in the microsomal metabolism, like the different concentrations, how they affect, and the uncertainty regarding the locus and modality of the ES interactions, etc. So we have been talking about the pull. Now, how is that pull? How did we get one slide? We have shown this here, the pull effect, thermodynamic pull that we talked about. And this slide also very clearly shows that when there is a substrate present, there's this thermodynamic pull. Now, this is, we are talking about the peroxide in the reaction system. And when you add the substrate, which is the hollow squares, so what do you see? At most of the places, what do you observe? You observe that the peroxide levels are actually down, actually less. Why is that? Because once the substrate comes inside, they're actually being taken up. So there's this thermodynamic pull which is actually driving this whole reaction. So that's why you get this kind of an observation here. Now this has been shown in different reaction systems, like it's just a test tube reaction here. In here you have a bacillosome system, and then you have the CPR system that we actually talk about, and then the reconstituted systems as well. So almost everywhere we have a kind of similar observation. Now, yeah. So this is an interesting study where, what do we observe here? We talked about the thermodynamic pull, right? Very evidently, when we look at this data, we are actually talking about CYP2C9 reaction, okay? And CYP2C9 metabolizing diclofenac, giving you 4-hydroxydiclofenac, which is the product. So this is the standard reaction. And when you add other enzymes, like you have SOD, superoxide dismutase, as we know of superoxide dismutase, it actually works on the superoxides, and it can actually only work on superoxide, and it can take that away. But still, there are other DROS species that will be available. So because there are other DROS species available, you still observe some formation of the product. And when you talk about catalase can work on peroxide. So still you have some other DROS which it cannot work on, and that's why you still get some level of, the formation of the product. CP also the similar case. But when you talk about HRP, CP is a little, hydrophilic kind of. It stays in the aqueous phase. So you observe this kind of you still observe reactions over here. But when you talk about HRP, it is a little hydrophobic, and then it takes away all the DROS. It can utilize all the DROS, and because it can utilize all the DROS, it does not allow cytochrome to actually hydroxylate any of the any molecule of diclofenac. So we don't observe any product formation over here. It's all driven up. It's all taken to a different direction. So it can only happen when there is a diffusible radical which is going out and doing the reaction. Otherwise, if it is so very active site mediated, this kind of observation is not possible, because the substrate will go and bind there, and then there will be hydroxylation, and you will eventually get the product. Some or other amount of product you will definitely observe. So we can also see here this ascorbic acid, which is again a soluble form, and then you have the palmitate attached to it. So palmitate is again hydrophobic. So when you add the hydrophobic moiety, there is inhibition, quite a lot of inhibition observed. And when microsomal systems are at the membrane, the reaction happens in the lipid phase. It's kind of the lipid phase where the reaction actually happens. So a lipid phase modulator, inhibitor works much better here. Ascorbic acid can also do the job, but then if it is in the soluble state. This is a classic piece of work here from where actually Sir had observed one of the starting points from where the ideas erupted. Now, what do we observe here? There is this reaction system where you have cytochrome, the enzyme, the P450, and the substrate inside a membrane, and then it is separated from the P450 reductase. Now, P450 reductase, according to the classical mechanism or the erstwhile mechanism, needs to bind to the P450. Without that, the electron transfer is not going to happen, right? So what if you separate them? You put a membrane between and you separate them. We are not going to expect any reaction if the erstwhile mechanism is actually correct. But then, interestingly, we not just only observe reactions of almost the same level, but we observe very specific reaction. 4-hydroxyl diclofenac is the product, and we observe that even when you have put the whole reaction in the tube. Why does that happen? The DROS that is generated, the DRS that is generated here by the CPR diffuses through the membrane. It can comfortably get there, and the reaction can proceed. This very clearly shows that the reaction is not driven only after binding. We're not saying binding doesn't happen at all. No, it's okay. It might happen, might not happen. Secondary. But what is driving the reaction? The reaction is driven by the reactive species, the diffusible reactive species. So it diffuses through, right? And then it gets inside there and the reaction happens. So now, if it is happening through the diffusible species, next thing was to observe the structures of all these P450s. So we took a lot of P450 system over here, and we tried to observe what is their how they are, in 3 dimension. How do they look? And there was one common thing that we observe everywhere. We see here that actually, if you look at 2S6, and 2E1, you can also observe the red dot over here and the green dot. So this is actually solvent accessible. The proximal site where the cysteine actually holds the heme is the proximal site, and that is accessible to the solvent. Invariably, in almost all, except this chloroperoxidase, it's accessible in all the systems, right? And yes, heme is very accessible here in 2E1 and 2A6. That's, we have, variations because of that. But of course, just to understand at least the diffusibility, the chances of things getting diffused is not only happening at the distal site, where the erstwhile hypothesis talks about binding of the substrate. That happens or doesn't happen as a secondary stuff again. Now, not only at the distal site, but also the proximal site is actually accessible to the solvent. So it can diffuse. If there is a ROS generated, if there is a DROS, it can diffuse through this way. And okay, I'll come to this, continue with the explanation here. Before that, that is one of the understanding that is required to understand the role of DROS in the reactions that it can carry. And then there was another big question. When you talk about, I told you that P450s are very promiscuous. They can metabolize a lot of substrates. Now, not just a lot of substrates. They can metabolize substrates of different topographies. Now look at this. We have small molecules. We have this big a molecule. We have much bigger a molecule here. All of this can be metabolized by one CYP. The example taken over here is of CYP2C9 and CYP3A4. This is the diversity of just CYP2C9. We can't just imagine this. If you take the classical understanding of enzyme kinetics itself, what do we know? We know about either the lock and key model that was given by Fisher, or we know the induced fit model that was given by Koshland. Now, lock and key definitely doesn't fit here very clearly. It cannot. It cannot explain the binding of such very topographically, varied substrates. Not possible. But when you even take into account the Koshland's induced fit, even that cannot allow the binding of a molecule like trabectedin or retinol in a place where it is going to bind amitriptyline. It can probably give you some space for this, but it cannot just unfold completely and allow trabectedin to get inside the active site. Not possible. But then trabectedin gets metabolized. How can it be? The classical understandings cannot explain. So, what can actually be happening? This is the plane where this is the heme, and it can take different oxidation states, and then it can, have different kinds of the oxygen, like you have oxo, peroxo. All these states are okay, possible. But then what else do we observe here? We observe different channels that are open, that can, from where you can actually access it. And if you look at that, if there are channels here, then, and taking into account Murburn, if there is a DROS release, DROS generated, it can be released and can be taken up by a substrate because of its thermodynamic pull at the place where it is binding. Okay, so it can be here, it can be in the active site, it can be somewhere here, one opening of the channel, then there can be this one, or it can be the proximal site. It can be either of it. So that's what is shown over here. Like you have trabectedin. Trabectedin cannot bind inside. It cannot. And we have seen this. With docking studies also we have seen it cannot bind inside. It cannot get inside. So, but it binds on the surface. And where does it bind? It binds very close to the channel. It doesn't bind randomly anywhere. It binds very close to the channel. There, it has a very high probability of undergoing a reaction. Now, this is what sir was talking about yesterday when he said Mur zone. So this is the Mur zone where the reaction can actually happen. The DROS comes out, and there is a very favorable chance of finding a ROS that can metabolize substrates. right? So going ahead, taking into account that, okay, fine, DROS is now, we have seen DROS can do this and DROS is doing it. Yeah, sorry. So let's get back here. So we have been seeing, fine, very clearly that it's the requirement of the ROS, the diffusible radical oxygen species. So if that's the case, then do you really need the partners that we have been talking about in the way they have been presented all the time? Exclusive partnering, like if you take P450 CAM has its own redox partners, the classical understanding. We changed the classical partnering of P450 CAM over here. And then still we can get the reaction very comfortably. We change the substrate. If you change the substrate, you cannot expect the reaction to undergo if it was a binding based mechanism. But if it is a diffusible radical driven reaction, the reaction can still happen if the substrate is present in the Moore zone. That doesn't mean that you can have any XYZ P450 for XYZ any substrate. No, that's not what we are trying to say. You need a Moore zone for that. And a Moore zone depends on several factors. Of course, my talk is limited. We cannot go into that right now. But then, yes, we have seen very clearly that when you are switching partners here, you have 3A4 reaction with coumarin, which is not the substrate actually for 3A4. That also can give you the product. And when you have P450 BM3, which is a bacterial P450 system, you allow that thing to metabolize coumarin. Even that can do it. And when you talk about CYP2C9, even that can do it. Maybe the efficiencies will change. It will change because there are a lot of other things that are driving it. There is a thermodynamic pull. There are other factors also, which will affect it a little bit here and there. But then the reactions can happen. So it is not absolute one-on-one binding in the active site. Only then the reaction can happen. No, it's refuted here very clearly. Yeah. And on top of everything, to just show that it is actually radical species, diffusible species, this reaction, you don't have anything. You just have an enzyme, no redox partners as such. There is no CPR, nothing. There is only superoxide. Superoxide is the DROS, one of the DROS, right? But it can alone do the reaction. This system alone can give you the product. So very clearly, this gives you a very strong proof that it is the involvement of ROS. It's not just binding and you need a partner, specific partner. You actually need ROS, not the partners. We had taken this further, and here in this particular slide, what you are observing is the case of benzbromarone, one form of benzerones, which was reported to have nanomolar levels of Ki values, inhibitory constants, which we could not buy because when you talk about inhibitory constants, it's actually Ki value. It's actually it's a binding based value. It's not just like IC50. IC50 can talk about reducing the rate of reaction. But when you talk about Ki is actually about binding of the inhibitor to the enzyme. Only then you can get that value. And when you say nanomolar levels, you're actually bringing it lesser than the amount of enzyme that you have provided over there. So even that is lesser than 1-to-1 binding. This cannot explain the kind of inhibitions that benzbromarone was claimed to have. So we took up that thing and then we observed, we did reactions with, and then it was also said that it is a specific inhibitor of CYP2C9. So we took up different systems. We took up CYP2E1 and other enzymes also. And we saw that no, they are not inhibitors of just 2C9, they are inhibitors of other P450 systems as well. Plus, there is very interesting observation over here. When you look at this, we have, this is the concentration, like you have 100 micromolars of the inhibitor here at this, in this row, and then 1 and then 0.01, that's much lesser over here. So if you talk about a binding based inhibition, this row should have the maximum inhibition. But what do you observe? You also observe, like you look at this particular case, you see here the inhibition is higher than what you observe here. How can you explain that? And that's not just one instance. There are other instances as well, like here. So here, so how can you explain that? If it is binding based, no, sorry, it cannot. And the non-specificity of the inhibition by benzbromarone that I just talked about is shown here in 2C9 system. It can inhibit 2C9 system. It can inhibit 3A4 system as well. 2D6 also, 2E1 also. They do not really have the same active site topology. So you cannot imagine, you cannot think of it binding to all these enzymes in the same way. It cannot be an inhibition just by binding. Yes, if it is DROS driven, and if benzbromarone can eat up the DROS and not allow the enzyme to proceed further for the reaction, then you will observe an inhibition. And that's something Murburn concept helps this to understand. Now here we see the ability of lipophilic molecules to inhibit much better than the soluble ones. That's again because P450 is a lipophilic. The reactions happens in the lipid. So that's why we see lipophilic molecules to act much better than the soluble ones. So what do we have here? We have seen that substrate diversity. We explained the substrate diversity, which the classical mechanism doesn't address too very well. And then there are so many other effects like secondary oxidations, kinetic TIE, kinetic isotope effects, then oxygen species binding to CYP, and then also about the electron transfer from the CPR. We have seen that no, the binding is not the protein-protein binding that is always advocated is not really required. So all these things, all of this observation that we have, the different observations which were not actually explained very well by the classical mechanism, Murburn has the ability, it actually explains all of them very cleanly, very clearly. Yeah. So here we see the role of, we have cytochrome C with the CPR, and then we see when you add the inhibitors, like you have cyanide and you have even copper ions, which can all of cyanide, we call them as binders and all. But yesterday itself, Vivian had talked about it. It must be clear that it's not actually a binding based inhibition. So as the ROS which it takes away. So we see copper ion also having the ability to do that. So, and then we have the effect of vitamins and some of the derivatives of the vitamins, which is like, we talk about this particular graph where you have a soluble form of the vitamin C, and then the lipophilic form of vitamin. So the lipophilic ones are not showing a lot of difference over here in the with respect to the inhibition of cytochrome, which is a soluble system. So when you put a soluble enzyme, soluble vitamin, you can see the inhibitions, the variations in the activity. Now again, what does that actually imply? It implies it's not really binding, because the system has to be present. I mean, the inhibitor has to be present in the actual zone where the reaction is happening. And if it is if the reaction is happening in the aqueous system, it has to be the molecule has to be hydrophilic, or if it is happening in the lipid system, then it has to be lipophilic, right? Yeah. This is a very interesting observation. Like when you look at the concentration of an inhibitor, azide is an inhibitor, known inhibitor for the people with the systems. Now, what do you observe here? We are going down with the concentration here, and then okay, we observe sudden activation at some position. Now, how can we explain the sudden activation? Again, the binding cannot do that. And then you again see a sharp dip. Higher concentrations, of course, there are inhibitions. But then there are other instances also where we have seen no inhibition at the higher concentration, and then there's an inhibition at the lower concentration. So if there is no inhibition at higher concentration and inhibition at the lower concentration, that again cannot be explained because of binding, right? Like you observe here, like there is an activation rather, right? And then it comes down here, it lowers down when you are lowering the concentration. So, and this, okay, fine, this is just the reaction, but here, the spectroscopic data that we see, the spectrums will actually show you if there is a binding happening or not. If there is a binding happening, there will be some shifts that we observe very clearly. But even with 100-fold excess of the, 100-fold of the inhibitor present in the system, you don't really observe any shift. So that shows there is no binding. There is no binding as such. Yeah. Now we are looking at a slide which talks about the hormetic effect. And what is hormesis? Hormesis is basically when you go down, you lower the concentrations, you observe some activity. Homeopathy talks about that kind of stuff, but the concentrations that homeopathy actually deals with, it becomes doubtful whether there is any molecule in that or not. So, as Sir said, we are not really advocating that part. We haven't gone into that kind of research, but yeah, something like what you see in homeopathy. So, here, when you look at the different concentrations of azide that is given to a growing system, you have Bacillus megaterium, gram positive and a gram negative system, which is E. coli. You do not observe effects on the gram positive, but then when you look at the growth, effect on the growth of the gram negative bacteria, it's very evident here. Like you have micromolar concentrations. This is the when you are having millimolar concentrations, there is a lot of inhibition, and then when you go to micromolar, it kinds of normalizes, and then again at nanomolar level, it takes a dip. So all this is very weird. But even more weird is when you go a little more down, you go to picomolar levels. What do we observe? You don't observe the 100% growth. We observe even beyond that. That's an activation. That's the kind of hormetic effect that we're talking about. Picomolars are much lesser concentrations. The control reaction rates are shown on the right-hand side. You don't observe such kind of thing over here. Yes, of course, at the millimolar level, you have a very sharp inhibition. Apart from that, no inhibition, no such activation either. There's not, even if we have observed a little bit, I wouldn't call that to be very significant for the things that we are understanding. Yeah. This is again a very beautiful piece of observation where you have two systems. We are talking about one enzyme here, which is the peroxidase enzyme. I told you we are going to deal much less with the peroxidase. We have HRP, phosphatidylase peroxidase, as one of the example, the model enzyme that we take. And HRP is actually used in a lot of things. Medical uses are also there. A lot of medical uses. A lot of detectors that we use has HRP. The ELISA-based readers. Right, all of them. The even the glucose strip that we use to check whether the blood sugar levels are okay or not, that also uses HRP. So, what do we observe here? We are seeing Okay, yeah, before we get into what we are seeing, now you look at, we are talking about this effect of cyanide over here, right? And cyanide, yesterday, Vivian has talked about it. Cyanide has a pK value, if I don't know, if you remember that, it's somewhere around 9.2. So when you get near 9.2, you like when you are at 9.2 or higher, you will get better inhibition. So when you look at pH 7, now here an observation, when you look at pH 7, the substrate and the enzyme, take both of them as a couple here, and we have HRP-ABTS even here. Now when you look at the IC50 that is observed at pH 7, it's still high. And then when you look at pH 9, it's very, very sharp, very high. And when you look at pH 5 again is coming at 194. So what is the kind of pattern that we are observing? 194, it goes high, and then it comes down much lesser. If it is dose dependent, you wouldn't observe this kind of a graph, right? Lesser at pH 5, higher at pH 7. I mean, actually speaking the other way around, inhibition more at pH 5, lesser at pH 7, and very strong at pH 9. That cannot be explained by a dose dependent. I mean, if it is, even if you take the ionic states, a gradient is not observed here. So the classical understanding of inhibition does not explain this. And further, Vivian has actually talked about it yesterday. So not spend much time over here. One very strong observation, this one, let's talk about this, where you have pyrocolol as the substrate, and then we are changing the concentration of the substrate. Here you have 2 millimolar substrate, so more substrate. To inhibit that, you need more inhibitor. What is the IC50 we are observing? We are observing 10 micromolars. Lesser substrate. You should need lesser inhibitor, but you need actually more over here. Just the ulta, just the opposite. Here there is no difference. How can that be? You don't have a difference. This is 10, much lesser concentration. This is 0.2 millimolar. This is 1 millimolar. But eventually you have the same values of IC50. 193, 194. You take the error by it's under that consideration. It's almost the same. It's the same value. Cannot be explained again by binding based mechanisms. So we have seen a lot of different observations. Like we just saw hormesis when you have lower dosage, and then at lower concentrations you have higher activity, hormesis. That can be again explained by the Murburn concept. And you have the idiosyncratic responses, where under certain conditions you get weird observations. Like this cannot be just explained by a gradient or some dose response mechanisms. And then all the other like zigzag curves that we have seen, multiple peaks, all of this can be better explained with Murburn concept than it can be explained by any binding based mechanisms. Thank you. Questions, please.
Yeah, thank you very much, Dr. Abhinav Parashar, for very well explaining the examples and experimental data on the comparing the Murburn effect versus the conventional effect on the CYPs and dose responses. Because as we all know, drug metabolism plays a very critical role in determining efficacy, safety, and therapeutic outcome of medications. And CYPs, a few classes of CYPs especially, they are the major enzymes involved in drug metabolism. So with a lot of examples, he explained how these dose responses or the variations, all this can be explained by Murburn effect. So now the session is open for questions from the audience as well. Yeah.
Please. Sir, there is a question there. Yeah, you are audible. Please tell me.
He was expected, yeah. Yes, sir.
Thank you. So, really, rather than asking question, I need to complement something because I work with the drug metabolism in live animal. Okay, there also we get conflicts like this. As you explained, the hormetic response, everything, but we are unable to explain it. Actually, we were thinking that it is because this concept is very new to me. So only last month I got when Saras invited me for this. Then only I came to know about this theory. Okay, then one thing I felt is that say when you are talking about the ROS, which is mediating, can you use any quenching mechanism for ROS and directly find out whether it is really due to, because in many places what you are saying is that it may be due to, because the classical theory doesn't explain it. You are telling that this it may make the Murburn theory holds better. Okay, so if that ROS can be done, block. The problem with my mic. Oh, okay. Have you done any experiment like this? Because I have not done any in vitro experiments. I have do in vivo experiments. That is my first question.
Stay here. Okay. Yes, sir. We have actually shown the results of. Yeah. Okay. The slides are not here, sir, right now. Yeah. Yeah. Here also we have done the same thing. Like the quenching of the ROS that we are talking about ROS mediated mechanism, right? So sorry. Sorry, yeah.
Okay.
So if you look at this is the basic basal reaction, right? CYP2C9 is the microsome that we are dealing with right now over here. And when you are adding a quencher of ROS, right? So SOD is a quencher of ROS. SOD is superoxide dismutase. It will quench the ROS. And it will quench which ROS? It will quench superoxides. Then we have catalase system. Catalase is again a quencher in this particular sense. Of course, it is an enzyme, but it's also a quencher. So it will quench H2O2, peroxides. And then we have HRP. Chloroperoxidase can also do the same job as that of what catalase is doing. But then when it comes to HRP, which also has a hydrophobic nature a little bit, it can quench DROS of different kinds. So that's why we observe a complete inhibition. So here, because
understood.
yeah.
Yeah, that I think now I was missing this slide. That's right.
Okay.
Yeah.
It's a soluble system, sir, over here, like ascorbic acid. We know ascorbic acid is as a good modulator. It's a very potent redox molecule, but it will work in the aqueous phase better. No, here we are dealing with CYP2C9, which is a lipophilic system.
Okay.
The reaction is happening over
So,
there. And when you modulate that, you change it. Like we have ascorbic palmitate. Now we have a palmitate chain over there. It can go inside the lipid phase and then it can inhibit that.
Okay.
So that's what I've been trying to project.
Just alphabet. There is no inhibition.
No inhibition. Understood.
Yeah.
Okay.
Site specific. Its place.
Okay.
There is.
Okay. Understood. That
SOD doesn't inhibit.
yeah. Okay.
My job was to see that. My job was to see that we minimize ROS production. during the lead drug optimization. So I reported to my professor that if we take out DROS, it doesn't work. So that's what is happening.
Okay.
They put ascorbate, nothing happened. SOD, nothing happened. But the point is to take the ROS scavenger where the ROS is produced.
Understood.
So he showed another system where you see cytochrome C getting the electron from cytochrome P50 reductase. And that electron reduction happens in the aqueous system. There you see the different thing. That's why I said yesterday, where the reaction occurs, at what time and in what amount the ROS is. That determines the outcome.
Yeah.
The idea that ROS in itself is bad or not involved is not right. This is the reason why we missed out all these observations.
Okay. Now, I noticed this disparities when we do the in vivo experiments. In the animals, when we do, we get the same aberrant responses. But we were unable to explain it why. Because that is a very composite system. It contains so many, all the cytochromes will be there, all the compounds will be there, endogenous compounds will be there when you take the case of animal. Because we take it from raw liver.
We published this in Dose Response. It is it's an American journal. Edward Calabrese was the editor there. And he was very excited. He said the exact same thing that you said.
Yeah.
We have been observing all these all the while, and we have been sidetracking it as, artifacts.
Exactly.
And now we have an explanation. So he was very thrilled. Exactly like you are.
And one more thing, sir. I think that question maybe. So can you derive equations for like this? The reactions like this?
That's wonderful. That's wonderful. We tried a recent paper in Biomedical Research International in 2026, Wiley paper. It's a Q1 journal. We tried to get the mathematical hang of it. As of now, we have got biphasic, zeroth order, depletion, all that mathematically figured out. But the complex phases, we will have to go a little more deeper. Wonderful question. Because on that alone rests our ability to predict. See, he showed some examples of hormetic things.
Yeah.
We started to model that. We couldn't get there.
Okay.
It's a very, very, very hot topic, because if we can give custom medicine, I have a friend who, when he sits on a plastic chair, he starts coughing. He sits on a steel chair, no problem. And people, they say, this is unreal. When you see our body as an electrostatic system, you can get to there.
Yes.
So there are physical, there are chemical, all this being sidetracked. Sometimes parallel systems of medicine, kind of by hit and trial, get there. So Murburn allows you a framework where you can mainstream all these things.
Understood.
I'm so happy to see somebody like that.
That's a really a wonderful thing that we never thought of such a possibility, that after coming here only I came to know this is one thing that is possible, and every theory it is to be modeled mathematically, then only it will have the fidelity. Okay, that is because acceptance will be. Okay. 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 Yeah. Sir. Yes, sir.
Because it has mathematical ground.
Yes, exactly. So you have that equation. Now, another thing is that in very of the small scales, what happens is the quantum phenomena that actually sets it. Quantum phenomena that are setting in. How you ever thought of such possibility? Because I'm another area I'm working is with the anesthesia. Okay. So in anesthesia, many of the phenomena we cannot explain by the conventional, the receptor theory. Receptor theory. And the second thing is regarding the mind or consciousness, what you call as the consciousness.
Mind, I don't know whether we can get there as of now. But then approaching mind is only possible with Murburn, because the classical theory is ionic. This is electronic. So that we'll talk about in the next session. That is neuro electrophysiology.
Yeah, exactly. That is, okay.
Next session.
Okay, okay. Okay, sir.
Yeah,
Thank you.
wonderful.
Thank you.
That's for you, sir. That's for you.
Wonderful.
Thank you, sir, for a very nice question. Sir.
It's from the field. So
Yeah. Very relevant. I think it will also help your research to grow further. Yes, sir.
Hello, sir. When we speak about microsomal enzymes,
Yeah.
so we understood that these present in the endoplasmic reticulum, but
Yeah.
there are non-microsomal enzymes which will be present outside the smooth endoplasmic reticulum.
Yeah.
So they are also making the drugs to metabolize in various reactions.
Yes.
May metabolize. Many are also microsomal enzymes are also.
Yes.
So can you provide reactive species which is not microsomal enzymes rather than liquid? It can be an interesting study on correlate with non-microsomal enzymes. And
We actually have done that. That's why you could see the connection between the peroxidase systems that we have taken HRP for that instance. Basically, we took off with HRP and catalase and chloroperoxidase systems. And then from the observations that we got there and the ideas we built upon there, we moved on further to validate that kind of understanding in the microsomal system as well. So it's actually
not microsomal, it's not.
I'm- non-microsomal. Peroxidases are non-microsomals. Horse-radish peroxidase, if you talk about, or if you talk about chloroperoxidase, you talk about, catalase system, all of them.
The point that you are asking is this kind of xenobiotic metabolism can occur not just in endoplasmic reticulum, it can occur in soluble enzymes and all that. So now the point is, the fundamental point that we want to see in xenobiotic metabolism is it doesn't have an evolutionary memory in your system, because normal molecules like carbohydrates, lipids, proteins, and all these kinds of molecules, they have evolutionary basis in your system. Their molecular topology is well known. You put a molecule like aspirin, ritonavir, sildenafil, any drug, it doesn't have. So Murburn offers the first basic scope for how either a polar or a larger molecule or a hydrophobic molecule, all those can be transformed and, gotten out of the system. So this Murburn process doesn't necessarily depend on endoplasmic reticulum stationing alone. It says that diffusible reactive species are generally produced in various locales. If you had come to my talk yesterday, I had shown that, whether in endoplasmic reticulum, whether in cytoplasm, any locale, whether in peroxisomes, whether in lysosomes, all that, extracellular region, it gets produced. So normal amount of ROS production, there is a sustained release. And then when that sustained release happens, these stochastic interactions with all these at different kinds of molecules at various sites can naturally happen. And that's how they get out of the system. So wonderful question. He was saying that.
Structures. These enzymes, because you show one drug with large structure, you know that the microsomal substrate cannot access inside.
Yeah.
It starts a particular active site. It can enter, right? Actually, you have doubt that because we cannot touch the inside structure.
Like if you look at the molecule that we are talking about here, I think you're referring to this particular molecule, trabectedin. It's enormous in size. It can never get through the channel also. If you get a deeper look in the at the structures, the structure function relationship, if you look at that, there's no chance trabectedin can actually enter. It's very huge. Even with respect to the channel, even if it opens up a little bit, because you can't expect the protein to completely unfold.
How?
That's what we have been talking about. The diffused, the ROS that is generated, let's say the ROS is generated at the, here. You look at this. Now you have a ROS generated here at this point, at the heme center. This is a simplified version of the enzyme. This is the whole enzyme. I mean, 1 second. Now, this is your enzyme here. Okay, this enzyme that we are seeing. This is the enzyme. Now you have a heme center. We know that this is, we are talking about P450. So we have a heme center. Now the heme center is somewhere here inside, just at the side here. And then at this point, you have the DROS that is getting, I mean, it's also held over there. That's another function of the enzyme that we talk about. But then let's assume the ROS is there, okay? Because it can, that heme center can activate the oxygen, right? So once it is activated, the ROS is there, or if it gets the ROS from the CPR that we have shown in the other example where we have separated the CPR and the SIP system and we have put a membrane. Either of that, you get a ROS over here that is held up, modulated in that particular region. And then once a substrate binds, comes here, like a molecule like trabectedin, you can imagine, since it cannot enter, it will be binding somewhere here on the surface. Okay. And the surface is not like blindly some surface. We're not talking about just randomly any place. It should be somewhere close to the channel. Now this channel that opens up, like you see here, this channel opens up here and very comfortably the ROS that is present inside can diffuse through. It can come and attack the substrate which is bound at the channel. It's like there's somebody huge coming from this, has to come through this gallery, but it cannot. What do? You have to send something to that person or whatever. We just let it go through this. It is diffusing through this. And if it finds that diffused molecule that we are sending at the door, and then it attacks, gets metabolized. That is what is, we are talking, that's what it is possible.
This is a very important question you asked. Actually, when I had gone to Gordon Research Conference in 2015 in US, it's a drug metabolism, the biggest conference, you know, Pfizer, Merck, all those people, they conducted. The question, Gabriel Cruciani, he's the supposed specialist to join. He's vacationing, so he's not joining. He just sent me an email yesterday that he has to be away. Gabriel Cruciani is the professor of drug metabolism and all that from University of Perugia, and he has a molecular discovery. He's a director of multiple companies doing this. He was making a talk there. So he was showing how these cytochrome P50s open up and allow these large molecules to go into the active site. He does all those high sophisticated computation. So I just stood up and asked him, don't you think this is all far-fetched? You know, he just stood there and he said, yes, I'm doing it because everybody expects me to. And that's the question that you have asked. Wonderful question. And then came Paul Hollenberg. Paul Hollenberg is a very seasoned scientist from University of Michigan. So he gave a talk where he mutated surface residues at different places, surface amino acid residues. So he saw that the SIPs failed to metabolize. So how do these surface residues mutations affect catalysis? So this Murburn explanation gives a very simple explanation. You see, that's what Abhinav has drawn there. There is a catalytic active site in the center, just above the heme. The molecule, it's like an elephant cannot come through this. But like he explained, the small ROS can definitely go there. The molecule can bind there, there, there. If this is the protein, right? That's what he explained. Wonderful. Nice explanation.
So, We want to.
You want to say?
No.
Yeah. 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 Please. Sir, please go ahead.
Since
Oh, that's enough.
since this is an ideas interaction, I'll just speak out aloud some of the thoughts which I got on the basis of listening to all of you. I'll focus on diabetes mellitus and the butyrylcholinesterase enzyme 1. You've done a lot of work on the mitochondrial pathological changes, the reactive oxygen species. I had been to one of the GRC conferences, a 3-day conference on mitochondria and their role in diseases. Maybe if you could apply the Murburn concept to the oxidative stress or the ROS and diabetes mellitus insulin resistance, that's a potential area in which you can work. That's one thing. Maybe some of the junior colleagues or the scholars can work on that. And second is, if you could just see whether butyrylcholinesterase is a murazyme. Yesterday, there was a deep learning analysis of this. There are 40 variants of this enzyme. And if you're interested, you could just look whether any of these fit into the murazyme. And you also showed that this drug metabolizing enzymes can fit into many pockets. So see whether butyrylcholinesterase or its variants can do that. And finally, we have data on raw data on butyrylcholinesterase sequences along with its variants. If some of you would like to do molecular dynamic studies, I'll be able to share it with my computer sciences colleague as well as you. These are some of the thoughts which I got as I was listening to you.
Thank you. Thank you. Thank you. Thank you, Dr. Sridhar. Thank you, sir. I think we are.
Thank you, sir.
Madam,
So thank you all. Thank you, Speaker Doctor, and then all the wonderful interactions. Thank you, Dr. Sridhar. So in the interest of time, I think we shall move to the next session, which is on two important, clinically important enzymes, lactate dehydrogenase and cyclooxygenase. I invite the speaker, Dr. Daniel Andrew. Welcome, sir, and over to you.
Thank you all.
Hello. Yes. Good morning to all of you. I would like to get your attention. I don't know whether you're, active, but when I speak, I want my hearers to be engaged and to understand what I'm saying. So at the outset, I would like to thank the organizers at CMFRI, especially Professor Murli, for the opportunity to discuss the work that we did jointly on two important enzymes in human physiology and medicine. One is lactate dehydrogenase and the other one is cyclooxygenase. So I go without much ado into my talk. Next slide. I can go here. Okay. 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 Fine. So I'm going to discuss these two aspects from published papers that we have. They are hypothesis papers. We are drawing on lactate dehydrogenase. We published this work in the Journal of Cellular Physiology, Wiley, where we said there are Murburn precepts for lactic acidosis, Corey's cycle, and Warburg effect. And we talked about how dehydrogenases, protons, and oxygen have interactive equilibria. Now, lactate dehydrogenase is an enzyme which reversibly, according to textbooks, it reversibly converts lactic acid or lactate to pyruvate. And it is a oxidoreductase class 1 enzyme which utilizes nicotinamide adenine dinucleotide, NAD coenzyme. So you can see that active LDH is a tetramer. You can see that in the image. And then there is a NADH binding pocket, about 10 angstrom deep pocket there is, which stabilizes negatively charged lactate and pyruvate because there are positively charged amino acid residues within the enzyme active site pocket. So here you have the NADH binding domain as well as the substrate binding domain.
Excuse me,
So-
we are not able to look at your screen. Is there a problem on my end or your screen is not displayed? We are able to hear your voice, not see your screen, unless the Zoom is disabled. Yeah, thanks. Now it's good. Yes, we are able to see.
Okay, these are the classical perceptions where we studied literature and we found out that the liver as well as the muscle have the same isozyme tetramer of LDH. You have LDH5, which is also called LDHM or M4. There are two LDH, LDHA and LDHB, which give independent gene products of A and B proteins. Now, LDHA is the muscle or liver LDH. LDHB tetramer, B4, is found in heart tissue and sometimes in RBCs. Now, you have got what is known as Corey's cycle. In the textbook, you know that lactate can be converted to pyruvate in the liver, but lactate dehydrogenase is supposed to be a reversible enzyme. The same isozyme you can see colored in blue, no, for LDHA4 or M or LDH5 isozyme is present in both liver and muscle cell. But then suddenly we had the question, if like in textbooks, it is a reversible reaction, why should lactate, which is generated in the muscle, go to the liver and be reconverted to pyruvate and then to glucose through gluconeogenesis, and that glucose comes back and this forms a cycle? Why should that happen? It is a reversible enzyme in textbooks. Furthermore, we found that muscle has 4 times more LDH than the liver. So it makes no sense why it should go to the liver. So with that, we started questioning how, LDH is involved in both Warburg effect and in Corey cycle. And if there is an alternate explanation for why, the main question you understand is, why does lactate have to go to the liver? So I will show you how we solve this issue. Not working. It's not. Okay, now it's working. So you see that lactate dehydrogenase, as the name suggests, it is dehydrogenating lactate. That is, lactate to pyruvate is so-called forward reaction. That should be the forward reaction. The name of the enzyme is tied to the name of the substrate it catalyzes. So LDH is a reversible enzyme according to textbooks with a delta G dash naught of negative 25.1 kilojoule per mole. And under physiological conditions, this should be something that the cell can, reverse, right? This is the textbook calculation from Leninger. Okay, and the equilibrium constant is like 10 power 4 to 10 to the power of 12. That is the range. But you can see here in this image, there are both steps 1 through 5, which show the backward reaction, right, of pyruvate to lactate, and the forward reaction of 5, 4, 3, 2, 1. If you go in that sequence where, the forward reaction is lactate is reconverted to pyruvate. The KD values are given there for you to see. The Michaelis-Menten constant for both these is given, and the equilibrium constant is supposed to be approximately 1. It can be about 10 to the power 4 to 10 to the power of 12. So I don't want to go more into this. Now, main question. There are two isozymes, LDHA and LDHB. LDHB is supposed to be mainly in the heart, and it converts lactate to pyruvate. LDHA is supposed to be in the liver or the muscle, and it converts pyruvate to lactate, according to scientists who have worked on this problem. And what is the mechanism of LDH action? It is acid-base catalysis. There is a direct hydride ion transfer which occurs within the active site, and that is how pyruvate becomes lactate. And again, lactate becomes pyruvate. It's all through acid-base catalysis because of histidine, and you have arginine, which is stabilizing the negatively charged ion of lactate or pyruvate. But if you look at the active sites of both enzymes, what do you see? They are superimposable almost. The position of NADH that you have, the position of oxamic acid, which is an inhibitor analog of lactate and pyruvate that you can see. The amino acid residues are all identical. Now, how does this differential recognition of lactate by heart LDH and pyruvate by liver LDH occur? There is no sense of that. There is no logic for that differentiation to happen. So the difference in topography or amino acid residues fails because the binding logic fails. Now, if you see the LDH can, according to the classical cycle, it is a very highly ordered thing. NADH must bind first. NADH or NAD plus must bind first. Only then lactate or pyruvate has to squirm past that and go in and be bound to the arginine residues. So that is a very, very, very ordered way in which it happens, right? So it's step by step by step. This binds, then that binds, then there is hydride ion transfer. But in the Murburn forward cycle, what we can observe or we can say is that ROS is usually generated in cellular systems, right? So when ROS is there, the positively charged residues in the active site can stabilize the ROS. So ROS can bind first. Sometimes NAD can bind first. So anything can happen. It's a reaction which is not, very highly ordered. So you have DROS. ROS can attack the lactate, which is bound somewhere on the surface. So in our docking studies with blind docking, we found that lactate is binding somewhere else instead of the active site pocket in a more favorable way. So this modified or Murburn forward cycle shows that lactate can bind somewhere instead of the active site and still be attacked by ROS. ROS is what converts lactate to pyruvate. It is not LDH. LDH is merely facilitating that by stabilizing the ROS. And ROS can have more prolonged lifetime. That is why we can say that LDH can be called a Murzyme. So now also singlet oxygen has been in photodynamic therapy and in photosynthesis and many other systems, triplet ground state oxygen is converted to a very reactive form known as singlet oxygen. That is also seen in, in our system, right? So that can also be involved in, attacking the lactate. So there are very different ways. It's not highly ordered. So both the forward and reaction and the reverse reaction are altered from the classical pathway. You have more stochastic bimolecular interactions in the Murburn purview, and DROS dynamics is there because of positively charged residues which can stabilize it. So we docked also the nicotinamide because we were interested in seeing what would happen. So now if you see lactate being converted to pyruvate, NAD plus should become NADH and H plus. So NADH is the product. If you see pyruvate becoming lactate, NADH is the substrate and NAD plus is the product. So we interchanged NADH and NAD plus with both LDHA and LDHB. And we found out that the orientation of NADH can change, right? So instead of the phosphate groups going in first, sometimes, instead of nicotinamide going in first, which should be closer to lactate or pyruvate, which is inside the active site pocket, it is binding in the opposite orientation with the nicotinamide ring coming out. And we found that this was more favorable energetically. So these kind of things suggest that, the system cannot clearly define the substrates, and that, binding can occur anywhere on the surface. Now you have a diverse molecules which are there, which can serve as inhibitors of lactate dehydrogenase. Most of them are antioxidants, very surprisingly. I do a lot of work on antioxidants. You have gossypol, you have so many other things like galloflavin, sesamol, all are antioxidants. How do these antioxidants mess with the system? Do they block the active site channel where nicotinamide, cofactors, coenzymes enter and where lactate or pyruvate enter? And is that mouth being blocked, and that is why we get inhibition? That is a question. But lo and behold, we even dock all those. So this is a diagram which can explain to you either with LDHB, where you have the forward reaction, that is lactate to pyruvate, or the backward reaction of pyruvate to lactate in LDHA, which is your liver and muscle enzyme. There can be different kinds of mechanisms that blockage can occur. An interfacial inhibitor can bind between the two monomers and give you lesser access to the active site channel, and that can be leading to inhibition. You can also have crypt blockers or oxamic acid, for example, which is known to go all the way in. So we have a lot of non-steroidal anti-inflammatory drugs, which are, known to be either selective. There are also compounds which can go and bind there. And we also have LDH inhibitors. I showed you those inhibitors like gossypol and CDMIM-QABA. I'll tell you about all these things. EGCG is a very famous phenolic compound, epigallocatechin gallate. All these are known to go and inhibit the LDH system. Now the clearest question we can ask is, why do they inhibit? Because they are very good antioxidants. Are they inhibiting LDH because of binding, or is it because of their redox activity? That is the key question. Now we took structures of various LDH A and B from different mammalian sources. Some active site channels were closed, some of them were open. Maybe you can say it is protein dynamics. But there is a lot of difference in the sequence also, right? When you align them and check, all of them are not similar. You can see that binding of these things, gossypol and all these drugs, happened at other places rather than the channel where you're supposed to see the binding. In blind docking, why did that happen? That's another question. So is it that there is dynamism in the protein, or is it like ROS is being scavenged by these molecules? That is the question. So now, the clearest thing is, why liver? Why should it go to the liver? The liver, according to the Murburn purview, is red because of high presence of cytochrome P450. Wherever cytochrome P450s are, where CPR is, the reductase enzyme, these enzymes produce a copious amount of ROS from oxygen. Oxygen is activated by these enzymes and ROS is released. So we found out that, I'll show you the other slide first. So when you have lactate, right, pyruvate and NADH plus H plus, you get lactate plus NAD plus. So if you see this equation, the energetics of this equation was miscalculated in textbooks. We rightly found out that the role of the proton H plus and its contribution had been ignored. When you put that into the equation, you get a value, delta G, of minus 476.4 kilojoules per mole. But if you go from lactate to pyruvate, you are reversing the reaction. The sign will revert. You will have plus 476. So the equilibrium constant is highly favorable, 10 to the power of 83, if you take the steady state concentrations of the lactate and pyruvate and the NADH and NAD plus, all these things, right? So you find out the equilibrium constant, it was about 10 to the power of 83, which means that this enzyme is going only one way. It is going from pyruvate to lactate and not vice versa. Because you need plus 476 kilojoule per mole, which is practically impossible to reverse even in a physiological system. But in the Murburn purview, because of these puppies of CPR and CYP, you see that their ability to produce a lot of ROS and the ability of drug molecules also to be there, right? Drug molecules are converted by this. You just had a talk on cytochrome P450 by my friend Abhinav. Now you see the contribution where lactate along with RH is a drug molecule, can be metabolized along with the drug molecule by the cytochrome P450 produced DROS. And the equation, the delta G that you have is negative 375 kilojoule per mole, and the equilibrium constant is like 10 to the power of 67, which approaches that what you saw, the impossible thing is possible now, because in the liver, along with drug metabolism and endobiotic metabolism also of our own compounds which we have, the liver is able to do this. That is why lactate goes to the liver to be reconverted to pyruvate, because it is a ROS-rich place. Inside the cells itself, lactate goes into mitochondria. Now, if you see the cellular ratio of lactate and pyruvate, it is like 10:1. Normal conditions, it is 10:1. That is why we can say that there is a 11th step in glycolysis, that is pyruvate becomes lactate. So lactate is produced very high, 10 times more than pyruvate. But in cancer, the ratio of lactate and pyruvate can be much higher, like even 100:1. Why does that happen? We have an explanation for that. So in the normal cells, on the left side, you can see that there is conversion of glucose to pyruvate and then to lactate. I told you lactate is the final step of glycolysis. The final product is not pyruvate, it is lactate. Now you have certain things here where there can be some Murburn in the mitochondria. Lactate is made, but it can be utilized as a fuel because it is electron rich when compared to pyruvate. There are electrons going from NADH to make lactate, right? So compared to pyruvate, it is electron rich. But what is the problem you have is the redox recycling is limited because you also have end product inhibitions in the normal cells, which are not cancerous. In cancerous cells, you might think cancerous cells are not, just metabolically defective. They are having higher overall redox throughput through the Murburn mechanism. We can say that while lactate is being produced, it can also go into mitochondria and become pyruvate. It can also, in the cytosol, there can be some Murburn happening if ROS is available. Cancer cells historically have more ROS than normal cells. And that is why the rerouting, it's not just, if you see Warburg effect, what is Warburg effect? Even when oxygen tension is high, you see that cancer cells are able to respire and they make more lactate because there is upregulation of LDH genes and there is more lactate production. But that lactate cannot enter the mitochondria too much. It is limited. So it has to go out into the bloodstream and go into the liver. So now, metabolic rates are higher in cancer, ROS is higher in cancer, which can support Murburn-based conversion. High cytoplasmic Murburn is there. And then there is, I hope, a way which we can discover where we can burn out cancer cells using Murburn principles and kill them rather than allowing them to proliferate. Now, I've already discussed this slide. There is some data here. So Abhinav and we were discussing, and his PhD scholar did this work. Credit to her, Nadia, Sheikh Nadia, and Abhinav is the guide. So here I want to explain this to you very quickly. You see, we showed that experimentally lactate cannot be reconverted to pyruvate. So you can see there is B, that is buffer, P is pyruvate, NADH. So what are we monitoring here? The rate per molar per second that you see is we are monitoring the NADH in the system, right? NADH, when you have pyruvate, what is happening to NADH? NADH is being consumed. So here you can see that, at different pH, you have both LLDH and DLDH. Two different forms of LDH were used. DLDH is bacterial LDH. LLDH is your normal, lactate dehydrogenase. You can see that at pH 7.4, the rate is very, very high. And at pH 9 also it is very high. So pyruvate is converted to lactate very readily, and you can see the y-axis, the numbers are quite high. But if you see this, here, you can see when we are checking lactate to pyruvate reconversion. Right here, what should happen if lactate is reconverted to pyruvate? NAD plus should become NADH plus H plus. The formation of NADH plus H plus is not very feasible. You can see the numbers are very low, and it is happening at pH 7.4 and 9, but the numbers are very low. It was 2000 odd when you saw the pyruvate to lactate reaction. That is NADH utilization. NADH formation is not so straightforward in our system because H plus formation is not thermodynamically very feasible according to the calculations that were done. Now, if you see, we go on, liver tissue was taken, liver lysate, and then NAD plus was added to check for NADH formation. And you have lactic acid, La is lactic acid, LT is liver tissue lysate, B is buffer, and NAD plus was added, which means it should become NADH, right? If you add LLDH and DLDH, both of them are doing something amazing. They are messing up with NADH formation. NADH formation is going down. If you take liver tissue alone, NADH formation is more. You can see the pink bars to the left. But when you add LDH, the moment you add LDH, NADH is disappearing. So LDH consumes NADH rather than forms it. Okay. So that is one story. And again, this analysis was done with KO2, which is potassium superoxide. The moment you add potassium superoxide in the reaction, and you can see that there is conversion of, NADH formation is there, NADH formation, which means that lactate is becoming pyruvate. But if you add LDH, the values go down significantly. Now, FTIR analysis was done to see the changes in spectra between lactate and pyruvate, and then you can see that lactate is not getting reconverted to pyruvate. GCMS was done to back up this observation using superoxide catalyzed oxidation of lactate, and Abhinav could show that, superoxide itself could do it, could convert lactate to pyruvate. But the moment you add LDH, right? You can see here LDH, if you add, it's going down. So LDH is messing with the system. That's why LDH is able to do pyruvate to lactate and not lactate to pyruvate. That's something that is very clear from the data. So you can see here in the GCMS analysis that superoxide can do it in the control reaction. You have both lactate and pyruvate at the same level, but then when you add KO2, right, you're seeing that lactate is getting consumed by KO2. KO2 itself can do it. That is support for liver being able to convert lactate to pyruvate. Okay, so now I'm moving on to the next system. If you have questions, you can ask me later. I have very less time. I'll try to finish in 10 or so minutes. So the other system is cyclooxygenase. Cyclooxygenase is very famous for what? Pain, right? The pain and inflammation is because of activity of cyclooxygenase. The natural substrate of cyclooxygenase is arachidonic acid, icosahexatetraenoic acid. We tried to reinterpret the structure function correlations of mammalian cyclooxygenase using Murburn concept. This is cyclooxygenase. You can see there, right, the active site channel for entry of arachidonic acid is very, very small. Certain crystal structures show that this can open up a bit more, but I'll come to the details. It is also a glycosylated enzyme, right? And it is monotopically inserted. Monotopically inserted means it is loosely bound to membranes. Cyclooxygenase is there near membranes. LDH is a soluble Murzyme, but cyclooxygenase is more a membrane-associated Murzyme. That's something that I should tell you now. So what does cyclooxygenase do? It takes arachidonic acid in 2 steps, in 2 different active sites. Cyclooxygenase has 2 different active sites. One is called the peroxidase site and the other one is called the cyclooxygenase site. Both of them are facing opposite to each other. The cyclooxygenase site is into the membrane. The peroxidase site is on the opposite side, away from the membrane and exposed to the solvent. So now you have interfacial chemistry here because membrane is there, solvent is there, the enzyme is able to do both, right? Wonderful enzyme. So you have arachidonic acid at a particular carbon 13, it gets attacked and there is endoperoxide formation, right? You have something known as PGG2. PGG2 is actually the product of cyclooxygenase site. It goes to the peroxidase site and then it becomes PGH2. So if this has to happen probabilistically, Murli sir did a calculation taking all the steps and the probability considerations, and for this to happen according to the classical way, it is a probability of 1 divided by 576. That is very difficult. So because you need this thing which goes in like noodles, right? It comes out like a hairpin, and we don't know how feasible this binding and dissociation is. We have not studied that yet. Later on, what happens to prostaglandin is you get prostacyclin, thromboxane, all these are involved in inflammation, right? Inflammatory mediators. That is why NSAIDs can block cyclooxygenase activity and then stop the process of inflammation. That's why we have NSAIDs. Now, I don't want to go into this, already explained the classical mechanism because there are so many steps. That's why these calculations were done, and that is a low probability thing for such a fruitful association to occur. This is the active site. Now, people say that, there are slight differences in the active site volumes of cyclooxygenase 1 and cyclooxygenase 2. Cyclooxygenase 1 is always seen in your system. Cyclooxygenase 2 is inducible. Whenever you have some inflammation or ROS or oxidative stress, cyclooxygenase 2 is expressed and made. So the minor changes in the active site volume, they say leads to the variability in substrate and inhibitor preferences. So therefore, there are COX-1 specific inhibitors, COX-2 specific inhibitors, and dual inhibitors of both the isozymes. So we tried to study that in both the peroxidase and cyclooxygenase site. We bound the molecules and checked whether there is any scope for differentiating the substrates of arachidonic acid, prostaglandin H2, and PGG2. Now you must understand that PGG2 is formed first. Arachidonic acid becomes PGG2 in the cyclooxygenase site. PGG2 has to go the other way around. What is the dynamics involved? Arachidonic acid has to come out of the membrane and go into cyclooxygenase site. That has to become PGG2. PGG2 has to come out of the membrane and go to the solvent. And from there, it has to come that side. So it's a very intricate mechanism that is proposed by people. But these are all lipid molecules. How well would they just come out of lipid, right? So there is a very interesting crystal structure of mouse cyclooxygenase, which I found, in which there is both productive and non-productive binding of arachidonic acid. In one case, the COOH group is outside. The other part, the methyl end, goes in. In the other one, the orientation is ulta. In the first case, A, they are saying that it is the correct or productive orientation. The other one they are saying is the non-productive or allosteric orientation. So there was one paper where they showed that all these molecules here, that cyclooxygenase is a dimer. Okay, understand that cyclooxygenase is a dimer. So there are two copies bound to each other. So they are saying that one is an allosteric site, the other one is a catalytic site. This also didn't make much sense to me. Why one should be allosteric and the other one catalytic? Different substrates have different substrate binding and preference for both cyclooxygenase 1 and 2. You can see according to the color code, there are efficient cyclooxygenase substrates, allosteric stimulators, inefficient COX substrates which are competing with arachidonic acid, and allosteric inhibitors. All this looks like a very elaborate story rather than, grounded in simple explanations. So we did a lot of docking of different compounds. I'll just tell you, I'm not going into the details. I'm just going to tell you that cyclooxygenase 1 specific inhibitors were found to bind to even COX-2. COX-2 specific inhibitors were able to bind to COX-1. That's why you cannot have a concept of specificity, because these inhibitors can go into both active sites. You don't have I think it is a myth of saying COX-1 specific and COX-2 specific. That's why in medicine, cyclooxygenase inhibitors, if you take COX-2 inhibitors, they can also bind to COX-1 and kill you. Right? There is that cross specificity. There is no specific binding. How can we explain that? So when we saw the protein, we saw that there are arachidonic acid entry channels. Tyrosine 385 is the most important residue in the active site, very deep active site of 25-30 angstrom. Tyrosine 385 is there and arachidonic acid squirms in and then it is converted to PGG2 and it comes out. There are so many channels. What is the role of all these channels? That is a question that nobody has answered. Why will it use only one channel? Why not other channels? Now coming to the last slide, perhaps, I'll show you some mercy and leave you. So in the Murburn view, ROS generation is always there in cyclooxygenase. You take any literature, you will see singlet oxygen, superoxide, hydroxyl radical. We say that all the NSAIDs you can see here in the right, they can mess with the DROS which is available rather than, having this thing going in and then something coming out and that going there and getting converted into something else. We are saying that it's a very simple thing that ROS, which is there, comes out into the lipid and it can convert. So that is what these equations are saying. Because of lack of time, I'm not going to explain that. We are saying that ROS can do it. ROS is good. That's all. I conclude. I'll be happy to take questions.
Thank you, Dr. Daniel Andrew, for the very interesting and energetic presentation and finishing it within the available time or considering the time shortage. So I think we can take one question and then others can interact with him maybe during the tea break.
Yes, sir. Yes. Sir, wonderful presentation. Sir, you have shown cancer cells. They are not going through Warburg effect.
They are going through Warburg effect.
Yeah. So,
Warburg effect, what do you mean by Warburg effect? That even in high O2 tensions, right? When oxygen is there also, lactate is being formed.
doubt which I have. You are also telling that ROS can be the reason apart from the Warburg effect. That's what I am.
Exactly. No, not apart from Warburg effect. I think you misunderstood. What is Warburg effect? It is there in cancer. What do cancer cells have? They usually have more ROS than normal cells, right? That's where the Murburn concept can apply. Normal cells, they cannot reconvert lactate to pyruvate. See, if you are taking a sprint, let's say you are running a 100-meter dash. By the time you finish, you get pain in your muscles. Why do you get pain? Because of lactate accumulation. Okay, now that lactate cannot be normally reconverted. It has to go into the mitochondria. Interestingly, in humans, mitochondrial LDH is a DLDH. It is not a LLDH. Okay, so you know that D and LLDH will operate differently. And also that DLDH is a flavozyme. It's a very different system. So in the mitochondria, why does it go there? Simply in the Murburn view, mitochondria is rich in ROS. People have calculated that around 10 million electronically modified oxygen derivatives are produced per mitochondrion per day. So therefore, being rich in ROS, mitochondria can reconvert. But how much of lactate can go into mitochondria? That is limited, right?
Because bird Billions of people they done PET scan, post-prone emission tomography. Because if you have open brain tumor,
I'm not disagreeing with Warburg. I'm not saying Warburg is not happening.
can be an artifact.
I think you misunderstood me. You can talk to me. I'll do my best to explain.
Yeah, so once again, let us thank both the speakers for the wonderful session. I thank all the members of the discussion forum and thanks to all the organizers for this wonderful opportunity. Thank you.
Thank you, madam. Thank you so much. I just wanted to say one comment to my student, Dani. Did you notice how he called cytochrome P450 and reductase these puppies? I really like that. Once you start working with some proteins, you start loving them. You start liking them. You start humanizing them. You start animating them. So I really like that. Nice thing. Yes. Laurent Jaeken, can we just have Laurent Jaeken give a message? Just a message. He was supposed to join yesterday. There was some problem. And today he has got a message. Can we read out what first? I'll read out. Do you have Laurent Jaeken's CV? Do you have Laurent Jaeken's CV? Otherwise, I can say Dr. Laurent Jaeken is a lifelong learner of cell biology. He has written a book. Laurent, can you hear me?
Yeah.
Good. So Laurent is a PhD product of Palladay, or Albert Claude or Palladay, I forgot. Albert Claude, sorry. He did his PhD with Albert Claude, Nobel laureate, in the early 1970s. And he has been teaching cell biology, and he has written a very, very, very enlightening book on coacervate and coherent mode of cellular life. And he has been collaborating, we have been collaborating together. Please put that on the, yeah. So today he'll serve, he'll start with Chaldakov's message first, before coming on to his message. So yesterday we had two chief guests. One was George Chaldakov, Professor George Chaldakov. So this is Professor Laurent Jaeken online. So he is a very illustrious and a very, very, very knowledgeable person. So before he comes to his message, he will read the message of Professor George Chaldakov. Professor George Chaldakov is the president of Bulgarian Society of Cell Biology. He is an MD PhD holder and associated with Medical University of Varna. He is the editor-in-chief of Biomedical Reviews and Adipobiology. So these two were supposed to be the chief guests starting off the meeting yesterday, but Professor Chaldakov had a problem. And now we have Laurent Jaeken, Professor Laurent Jaeken, Dr. Laurent Jaeken, reading out the message that Chaldakov, Professor Chaldakov, who I call senior BHF, see his degree. First degree he writes is BHF. It's brain and heart friend. He has got such a wide network, such a gentle soul, such a great constructive soul. So Professor Jaeken will go through his message and then come to his own message. Please bear with me. Please, sirs, please. Please, Laurent, please go ahead.
I want to thank Professor Murali Manoj for the invitation. And I will first speak in the name of Professor George Chaldakov from Bulgaria, who is the editor-in-chief of the Journal of Biomedical Reviews. And I think that like this, he started to know Professor Murali Manoj, because in 2017, Manoj was sending a very interesting paper, and I suppose Chaldakov was really struck with it because it was completely new. It bothered with everything. And so there were other papers sent to Chaldakov's journal. And next slide, please. Next slide, please. I think there are more than 8 articles from Professor Manoj in this journal of Chaldakov. And in these articles, they really reorient oxygen metabolism. It is like in the past and in the traditional way of thinking in biochemistry and physiology, oxygen, it has not very much work to do. It only has to react with the last enzyme of the electron transport chain. But in the Murburn proposal, the Murburn concept, oxygen is everywhere. And this was changing very much because by going to this new concept, we really come to the physical chemistry of biology, of cells. Before there was not a good physical chemistry. Here are 8, on this slide, 8 front pages of the biomedical reviews where they show work of Professor Manoj. It's a lot. If you have 8 times the front page. There was a front page also in adipobiology because it's in Murburn, it's becoming a very simple system. In classical understanding, in the adipocytes, the triglycerides are hydrolyzed, the fatty acids go to the mitochondria, and the mitochondrion, there is proton motive force, and then heat is produced by, they think that the proton motive force is dissipated. But in the Murburn understanding, it makes much more sense because when the fatty acids produce the NADH, the NADH is in the mitochondrion. It is oxidized by oxygen, and there is superoxide produced and other reactive oxygen species. And when they react with each other, one product, because these are all radicals, with an upspin, the other with a downspin, then the energy is dissipated and you get heat. So easy it is. I think the first of these articles is from 270, and it was really devastating for chemiosmotic theory. And the Murburn was proposed instead. There is a later article about this subject a few years later, and where really the question is posed, why do cells need oxygen? And you see there are two models. At the left, you have chemiosmosis, And what they tell is that the electron transport chain, the complexes, respiratory complexes, these are pumps. These are proton pumps. They pump the protons to the outside. Then you have the buildup of a proton motive force. And these protons, they want again to come in the mitochondrial matrix. And with this energy of this gradient, you make ATP. But that needs a lot of energy to make ATP. According to the reaction, ADP plus phosphate gives ATP plus water. Because you're in a watery environment, to add still some water by hydrolysis. It's not easy. But I just give two arguments to completely abandon the whole view. First, if you calculate for a very small mitochondrion or a very small bacteria, I saw mitochondrion in three-dimensional sections in series of only 0.3 micrometer. So it's so small that there is but one proton. How you could ever make a gradient with this one proton? How the many redox complexes should share this one proton? And then build a proton motive force, which is sufficiently great, large, to make ATP. It's pure nonsense. That's the left, to forget about. Then we come to the right side, and it's different. The electron transport, it's not a chain no more. It's just individual respiratory complexes. Each individual respiratory complex it was shown, can produce superoxide and other reactive oxygen species. That's known. Each one can it. If they give one electron from to oxygen, you have the superoxide. It's also logic that this respiratory that this superoxide is produced. It's logic because it's a radical reaction. These are the fastest reactions there are. And there is a huge, there is 150 kilojoule per mole. It's largely exotherm. So it should happen. And it is also shown that it is there. And then Professor Manoj made the in vitro reaction of ADP plus phosphate plus potassium dioxide. And you get just chemical without any enzyme. You just get ATP plus water plus hydrogen peroxide plus the large amount of energy. So why should ever a cell have invented a very complicated system to make ATP with a chemiosmotic mechanism if it could just make it like this? That's even possible by pure chemistry. And it's a pure chemical reaction. It's done in fluid. And you understand that if it is so simple that Chaldakov was impressed by it. And so there came a very nice friendship between Professor Manoj and Professor Chaldakov. Yeah, next slide, please. Then this year, in the beginning of the year, Professor Manoj was invited by his colleague in Bulgaria and was elected the honorary member of the Bulgarian Society for Cell Biology. Chaldakov also made a book on cell biology, a textbook, and he was so impressed by this new work and really the vision of it, so that he included this new concept in a textbook. So happily, there is at this moment already a good textbook. Thank you in the name of Chaldakov that I could speak here. I will now present my own presentation, and it tells that the Murburn concept is a really unprecedented revolution. It's difficult to imagine how deeply it penetrates in all aspects of cell biology. In 221, I wrote the book, The Co-Asservate Coherence Nature of Life, Fundamentals of Cell Physiology. It goes about the nature of life. It's not a hypothesis. It goes about things which were directly observed. It was directly observed that the cell is a co-asservate and not the cytoplasm, the fluid in it, is not just a diluted solution. If you put salt in water or sugar in water, you still see the water. And the ions and the molecules are dissolved in it. If you open an egg, you see that there is a gel coming out. You see it with the naked eye. It's not a solution which is coming out. If you isolate things, cell organelles, then you start to add a buffer, a fluid, and you dilute everything, and it may appear that it looks like a solution, but it's already treated. It's no more in vivo, the in vivo situation. So the cell is a co-relation, and the Murburn concept of Professor Manoj, it goes very well together. And I have been busy mainly to see how these two go together. And together they can completely replace the idea of ion pumps. You can really forget about ion pumps. The enzymes exist, and I believe for a while they will still be called pumps, but they do not work as a pump. It's completely different. Their mechanism is completely different. And what you see on this side, we are really on a new era in cell biology. And we are now in an era where science is really confused. Very, very, very confused. In modern cell science, on certain pages of textbooks, they proclaim that there are pumps. It's called membrane pump theory. The name is not very much used, but everyone knows about pumps. And they use the physics of diluted solution, the laws of Fick for neutral solids, Nernst for ions, Van 't Hoff for water. And this idea of pumps makes up the bioenergetic of the cell. The bioenergetic, it is that solids which can pass the membrane, they go through it passively, and then the pumps must compensate it, and they should work all the time, non-stop, because the passive movements don't stop. So the pumps must also come. But if you calculate it in very good experiments done on liver cells, but also on coli bacteria, then you find that these pumps consume much more energy than the cell can deliver. from their oxygen measurements. So it cannot work. It's a thermodynamic fiasco. But these things are in every textbook. In the same textbook, you find that the viscosity of the cell, of different cell regions, varies of over 1,000 times. How this is possible for a solution? For a colloid and a complex colloid, coacervate, this is normal. So the cell is a coacervate. And if you see, this is an internal contradiction in the same books, in the same university courses. If you look to Wikipedia, you will read the same manner of thinking about many things, unless you ask for a good question. With AI, it's the same. If you ask a question about Murburn, you will get an answer according to Murburn. But if you pose a general question, the AI will give you a nonsense answer. With I will first say what you can do with coacervation to give you an idea that you don't need the pumps. In cells, the water is not ordinary water as it comes from a tap. It is polarized, it is bound to the protein matrix, and this water has very low solubility for sodium. So you don't have to pump to reach this situation of differential concentration for potassium. For potassium, it's different. It has a high concentration in the cell, and if these proteins bear negative charges, and everyone knows that on average, proteins have more negative than positive charges, and there are very many techniques of proof that potassium can bind to some of these proteins, not all, and this can explain the potassium concentration, the high. So you don't need pump. It's with these two mechanisms, and there are other, and Murburn also has his parts in this equilibrium, but these are all equilibria. If these equilibria are disturbed, they will return to the equilibrium afterwards. That's what happens in an action potential in muscle contraction. It is disturbance of the equilibrium. Then it goes, it should be restored afterwards. And then at that moment, it needs energy, but not continuously. It only needs energy to restore the resting state, let's say, to get again to a relaxed muscle or to come again to a rest potential. That needs energy. But that's not the only thing. I have been studying coacervation from 72 till I was writing my book. And I brought together an enormous amount of arguments in favor and falsifying chemiosmosis and membrane pump theory. But those people who bring a coacervate view, there are a few who produced an alternative theory. In these alternative theories on coacervation, there is not very much about redox. And I have been working in a laboratory with Albert Claude, where there was also Professor Goffoni, he worked on oxidative phosphorylation. And I daily, every midday when we eat together, there were huge discussions about this, but no one comprehended it. And this is still the situation today. It's written in books, but no one comprehends it. I didn't comprehend. I tell you something. I once, in an experiment, I got enhancement of oxidative phosphorylation by adding dinitrophenol, a well-known inhibitor of oxidation, and I get enhancement, and no one comprehended it. Also, I didn't not. So it's not published, but now it's easy with the theory of Murburn to explain that. And it's in some articles it's written. I give you another example. There are old studies on mitochondria, on mitoplasts. So the outer membrane is eliminated on mitoplasts, and they add a detergent to destroy the membrane, and they could still make ATP. Impossible with a chemiosmoke, that the membrane should be, inner membrane should be really tight. Dus what happened in all this discussion? They forgot just one thing, and that is there exists another reaction, the one of Manoj. ADP plus P plus ROS gives ATP plus water plus H2O2 plus the energy. So it works automatically. You cannot escape from it. It happens. It just happens. You have it. Manoj also found that complex 5, the F1, F0, F1 ATPase, has also Murburn sites. So you need Murburn, and with Murburn together with coacervation, you do no more need pumps. I will thank you for the invitation to have the occasion to speak here, and I would tell the following, that your country in India can be proud about having such a scientist of that exceptional level. You are also with enough young students. It's a young country with a young population, and they are good scientists to bring about the change of paradigm, to go to the Murburn, to abandon the membrane pump theory, abandon the chemiosmosis. It should start somewhere. And in the West, it's really difficult to oppose to all these established people who want to continue what is already established according to them. There is a huge job because the textbooks must be changed, the courses must be changed. There may be needed a reinterpretation on perhaps each page. It's huge. The metabolic maps, you have very nice metabolic maps from Boehringer Pharma, for instance, but there are real large parts in redox which need to be changed. The scientific and medicinal enzyme, it will make a really large jump, because now ROS are understand as bad things and also as signaling things, but now we have the real function. So we can, it's all becoming logic. It is thermodynamically sound, it is kinetically sound, it is structurally sound, and it is evolutionary sound. Because if you look to those people who study the origin of life, they say life is a coacervate. And if you see to modern light, suddenly this coacervation disappeared and it became a diluted solution. How crazy is that? So one thing I say, remain open-minded and investigate. Thank you for your, to the audience.
Thank you, Laura. Thank you. Thank you so much. Thank you so much. We are already getting late for the next session. The tea break is already in. It was very, very, very insightful. Professor Laurent Jaeken has already been working on this. He has opposed the classical membrane pump theory all his life. And with what's his name? Ludwig
Edelman.
Ludwig Edelman. He published a paper which says challenging membrane pump theory is not permitted. Why? Multiple novels have already been given. The whole establishment needs to come down. That's what Professor Laurent Jaeken was highlighting. You know, it's violation of basic common sense that in spite of having refuted time and again, basic thing that he pointed out, cells are not solutions. They are co-servates, but we need to treat them as solutions. Such things. Now, Murburn concept and co-servation can completely replace whatever they have been saying for the last 70 odd years, you know. So thank you, Laurent, Professor Laurent Jaeken. So we will, I think, anybody wants to ask Professor Laurent Jaeken something? Yeah, I think the organizers are a little, pressed for time. So thank you so much, Laurent. Thank you for coming on. It was a wonderful talk. Thank you so much. Thank you so much.
Thank you also.
Next session will be in room number 201, Echo, and you can take lunch token after during tea time from the registration desk. So next session will be in room number 201, second floor. Okay. Sir, session is closed, sir. Thank you, sir.
Laurent Jaeken, Laurent, you can disconnect. I think we're breaking for tea. We'll rejoin. Please, Mahendra, Dr. Sridhar, Tofia, Sukumar, please, we'll join in about 15 minutes for the next session. Yeah, thank you.